Hello, and a very warm welcome to everyone joining us for this SelectScience webinar titled Developing the 5-Minute Point-of-Care PCR Test, a Technology Revolutionizing Molecular Diagnostics. My name is Cameron Smith, I'll be today's moderator, and I'm delighted to be joined by our guest speaker, Dr. Andrew Anyakwo, Head of Reagent Development at LEX Diagnostics. During the webinar, please feel free to submit any questions you may have. To do this, simply hover over the speech bubble icon to the left of your screen and click to open the submission tool. Throughout the presentation, we will also be circulating some poll questions. If you could please take a moment to submit an answer when prompted, it would be greatly appreciated. Thank you. So without further delay, I'm delighted to hand over to Andrew and would like to thank him once again for presenting to us today.
Thank you very much for that introduction. A huge thanks to the team at SelectScience and LGC Biosearch Technologies for organizing this webinar. I work for a startup firm, which is a spin-out of a major diagnostics technology company based just outside Cambridge in the UK. We were actually founded in 2020, and I'll spend the next half hour or so discussing our 5-minute point-of-care diagnostic test for flu and SARS-CoV-2. The word LEX actually stands for low energy extreme, and the initial concept was born on the existing point-of-care flu market. I'll be interchangeably referring to point-of-care as POC in this presentation. So flu POC has been around for about 10 years, and our original plan was to bring molecular diagnostic accuracy to this market. But this has now pivoted to an influenza COVID differential since then. So the graph on the screen shows a combined 2018 and 2019 data. So the purple is 2018 and 2019 is in orange. From a survey, this was actually taken from a survey in the Journal of Urgent Care Medicine. It shows flu coming out top of the 5 most performed rapid tests in urgent care. Addressing point of care in a bit more detail now. It's a form of testing in which analysis is performed where healthcare is provided as close as possible to the patient. The justification for performing a POC is usually when there's a very clear benefit to a fast result, as on a per test basis, they can get quite expensive. However, there is a limit to resources in central labs, and professional clinicians may be less skilled than laboratory staff. This places greater demands on the design of the test manufacturer. Current tests do have a few shortcomings, and which we'll get over in the next 3 slides or so. Ease of use is a very key requirement of point-of-care tests, and it's a current shortcoming of many of the molecular tests out there in the market today. Because they do require manual swab elution and handling of the swab, this introduces variability and sample collection between different users. The strategy of LEX is to automate the entire thing, so we introduce an automated solution which reduces the complexity of manual swab handling. So just describing the user workflow briefly is actually very straightforward from a user perspective. So once you swab the patient, the swab head goes into the cartridge, which we also refer to as the consumable. That gets swabbed off, closed, and inserted into a reader. 5 minutes later, you have your result. Now, the idea of LEX is the whole lab is on the cartridge. So at this stage, the clinician has basically done the job, and once again, we are actually taking human variability out of testing. Current point-of-care tests, so they're either low cost, if you refer to the antigen tests, which have less accuracy, or accurate like the molecular tests, but these are of higher cost. So low costs were actually very key in controlling the COVID pandemic, right? However, people who turn up to primary care, they do want accuracy, and LEX design is to fulfill the need for this high accuracy, however, at low cost. Unfortunately, the reimbursement constraints limit the use of this high-cost POC PCR test in primary care. Now, one might ask, we already have established POC tests that are good from swab to result in a 30-minute window. Why do we need to go any faster? To answer this, I'll give a little bit more context. So the current timing for primary care appointments is round about 10 minutes. In addition to the fact that current tests are good for 30 minutes, there is also the usual occurrence of having to reswab. If you've used point of care tests, it always comes up at some point, mainly from human error or machine error. And at this time, the patient appointment window might have passed and they would have left the building. However, with a 5-minute point of care test, there is a higher chance that the patient is still within that 10-minute time frame and a new test could easily be recommissioned. The greatest benefit of LEX's testing will be aiding the commissioning, performing, and discussing of test results in a single appointment, with the opportunity to do a reswap if anything went wrong, to further stress the error in point of care devices as a whole. We haven't got data for respiratory, so I've just pulled up data from the Abbott iStat because it's still in the same rushed environment. Okay, now the graph shows iStat error rates across 15 different sites. Now, if you look at the top of each bar, you would see that except one, the actual errors are actually from sample prep and the actual device itself. Now, this means that a lot of repeats would have had to be done in this instance. Staying in tune with the changing lifestyle of people in general, which means the public now do generally prefer quick answers to the issue of infectious diseases stemming from the COVID pandemic, the focus of point of care is increasing. Consequently, the heart of LEX is bringing extremely fast PCR in a true point of care sense. Over the next few slides, I'll go a little bit deeper and explain what we have achieved from a PCR and reagent development perspective. Firstly, it's very important that I stress that ultra-fast PCR is very possible. And LEX is building up the concepts of established and published principles. Now, in 2015, in a clinical chemistry journal article, now the work of Farah and Whitworth demonstrated extreme PCR speeds and showed that high-yield PCR is possible in less than 15 seconds. It turns out that if you shuttle DNA, of course, with the right amount of oligos and enzymes, between the right thermocycling temperatures, for instance, it is possible to evidence amplification of short fragments in less than a minute. Now, Whitworth's work has given us confidence that PCR can be done quickly, and LEX is confident that this can be done in a true point-of-care device. Now, the question will get asked is, how do you go about reducing this entire thing, this huge thing, into a small point-of-care device? It's worth stepping back for a moment to talk about the heating, as heating is the real core of LEX technology. Firstly, I'd like to put it out there that shape is actually very key. Now, this descriptive image shows a shape comparison between LEX and what you would refer to as a standard PCR reaction tube. Now, most of us will know this kind of shape because the traditional cylinder cones, they are actually not ideal for heat transfer, with liquid taking a lot of time to cool down. Each of these wells can range from a few millimeters to 3 millimeters tall. So when we look at how quickly they can thermalize, if you look at the temperature gradient, it takes after 8 seconds for you to start getting back to your anneal temperature, which are around between lower than 60 to 50 degrees, for example. This actually means that regardless of how quickly you can ramp up the temperature, you need to have a very long dwell time to ensure that the sample comes back to a uniform temperature. However, LEX is built on thin reaction chambers. So we have very thin liquid, meaning that we increase the surface area so we can rapidly ramp and we can cool. Following this core innovation, LEX is working with recognized reagent manufacturers, such as LGC, to optimize PCR reagents that will fit this new paradigm. If we want to go substantially faster, then, like I said before, we need to thermalize much more quickly. Just to drive home the point of low cost in this context a little bit more, this image is taken from one of our first prototypes showing our very thin PCBs in brown, which are relatively cheap to manufacture. Now, this enables us to keep the price of our consumable down, as our end goal is speed at low cost. Talking more about the practical assay workflow, the key driver of the LEX technology is really the consumable, which we also refer to as the cartridge. Going a little bit more in depth into the onboard lab, now this is our onboard lab, the consumable contains a swab pod, like I mentioned earlier, that sits in nasal swab. Once the swab is inserted, the lid is closed and inserted into a reader. Now, what happens in the reader is what I'll describe in a few minutes. So the consumable contains a buffer-filled blister solution that floods the swab pod once you hit start on the machine, right? So virus particles that are in the swab, along with other contents of the nasal swab, are then carried into the lysis chamber just here. Here, the sample is heat treated. Now, this does two things. It breaks down some of the inhibitors from the nasal swab, and in turn kickstarts viral lysis that leads to the release of RNA. The lyse sample is split into 4 distinct portions where it rehydrates 4 lyophilized separate PCR reagents. Now, each of these reagents will hold its own set of primers and probes, so we can detect both fluid types and a duplex SARS in individual channels, along with having a sample control channel. Now, the lyse sample, along with the reagents, go into the amplification chambers, and then we can begin thermocycling. And that happens just here, with fluorescence readout taken after each cycle, as it's done in every normal PCR. So, to talk about development in a bit more detail, our development has taken an iterative approach. I had the reagent side of things at LEX, and we start development of the LEX platform. So we basically buy in a standard live compatible qPCR mix, and we do our development on benchtop PCR machines, where we have set the thermal cycling to the fastest settings the machine can cope with. We do rely on design of experiments using JMP extensively to optimize the concentrations of novel compounds that add inhibitor tolerance and can also cope with ultra-fast speed. Thermal cycling is then done on LEX's own PCR platform, as this can really cycle quite fast, where we then put the reagent mixes that we've developed in-house through their paces at speed, and it all comes together in a full system development. Focusing a bit more on the LEX reagent side of things. Now, our Gen 1 product aims to target the respiratory viruses of flu and COVID, with the first goal being to retain clinical sensitivity with a reagent mix that is adapted for ultra-fast PCR. Now, in addition, this mix has to be suited to direct PCR. We want to eliminate the need for sample prep, reduce error, and manual extraction. And to make things even more interesting, the mix has to retain stability at room temperature. Now, the goal is to have the test literally sitting as close as possible to the clinician or end user, eliminating the need for any complicated or demanding storage requirements. Our strategy for addressing inhibition is basically down to what we can do on our consumable, our cartridge there. Now, in contrast to standard conventional POCs, we don't have any sample prep or pre-nucleic acid extraction. It's important to note that our buffer-filled blister holds a very small volume in comparison to the number of dilutions that happens as part of sample prep with other point-of-care devices. Our heat lysis has been optimized to be sub 1 minute, with buffer and PCR additives selected from both room temperature stable and lyocompatible components. Before any PCR happens, we do have to figure out a way to deal with the virus, able to lyse it, and whatever extra bits that has come off the swab. Now, our sample conditioning, we exploit the fragility of the envelope protein of the influenza virus. So both the influenza virus and the SARS virus, they do have this envelope protein that we can easily exploit by using a combination of thermal and chemical treatments. We've been able to reduce the time for obtaining RNA to less than a minute. However, right, the lack of subsequent cleanup means we have to deal with a lot of inhibitors, a lot more than previously possible within the molecular diagnostic PCR space. As anyone who embraces tough challenges, right, the first part of problem solving is fully understanding the problem, right? And you also have to take into consideration the worst case scenario in mind. Okay. So this table is an example of simulated respiratory secretions taken from a +1 2016 research article by Bosse et al. Key to also note that occasionally nasal swabs do contain blood. Now, this example and other sources of literature were used as a guide in contriving the worst case inhibitory system that would act as a base for commencing reagent development. Now the question becomes, when you have such little to no dilution of inhibitors, no cleanup, and including the fact that your RNA is complexed in this same inhibitory matrix, one would ask, is it ever possible to deal with RNA and PCR inhibitors at these high concentrations? From our development in-house, we've identified mucin as the worst culprit with inhibiting PCR from samples in the nasal fluid. Now, looking at papers out there, there are many successful strategies and examples of some that involve the use of certain enzymes. Now, this might be limited, they're not limited to proteolytic nature, right? So you could use trypsin, papain, and bromelain to cleave the cysteine-rich regions of mucin, or you could actually attempt to break the disulfide bonds in the same region. Other strategies have exploited the ionic interactions of calcium to sequester calcium. Now, when you do this, you increase electrostatic repulsion between containing oligosaccharides. However, this would need some sort of cleanup because these compounds do affect PCR. LEX's reagent strategy for dealing with the same sample inhibition was an extensive process of figuring out novel pathways that do confer mucin tolerance and also tolerance to other components of the nasal fluid, whilst protecting the active sites of DNA polymerase. Whatever we can come up with actually is quickly narrowed down to lyocompatible compounds, right? Which then form a complex mix of a stable product. Keeping in line with low costs to LEX as a company, right? And ultimately the end user, LEX reagents just have to be stable. In this regard, we have chosen lyophilization as a route to achieving this. And our PCR reagent development begins with lyophilization now, as opposed to the conventional method of completing PCR development on one instance before the addition of excipients and lyophilization following thereafter. Now, this means that we can introduce different components early on, we can optimize their concentrations, and we can monitor different key parameters of shelf temperature, chamber pressure, that would ultimately improve PCR performance over a long period of time. To ensure that our reagent development can keep up with the speed of the demand required to drive such a fast-paced startup company like LEX, our reagent production is fully in-house. Particularly, right, this ensures that we retain the know-how of all our mixes, meaning that we can react rapidly to the demands of optimization development to ensure our final formulation is coping with both PCR inhibitors and ultimately speed. Doing this, we closely monitor key PCR key qPCR readouts of CQ, amplification efficiency, fluorescence values, to mention a few, over different batches. Now, this gives us confidence with the amount of formulations we come up with. So when we see improved performance in a new formulation, we know that it's actual readout because we have extensive controls in place. The speed at which we have been able to get to this stage is largely due to the huge support we have received from the technical specialists in LGC Biosearch Technologies. Basically, right, when you start working with any new enzyme mix or any new component at all, it's very invaluable to understand what works and what doesn't work as quickly as possible. LGC have really supported LEX with custom projects to meet the demands of our PCR reagent development. And very, very importantly for us is their commitment to meeting our cost of goods at scale. This is key for us if we want to be competitive with the current antigen alternatives out there in the pork market. Now, this year, LGC launched a new RT-qPCR mix, the Rapid Fire LioFlex mix. This mix is suited to point-of-care PCR. The mix has been fully optimized for multiplexing. It boasts tremendous temperature stability, taking into considering the amount of development and the wet mix DOEs and JMP we do. It's very, very temperature stable. And after lyophilization, it retains stability for a long period of time. It is also glycerol-free, which is key to being lyocompatible. It's a 5x stock, which gives us the space to introduce various compounds that give us inhibitor tolerance at speed. Now, talking about some results. Basically, when we started this journey, the key was benching the best inhibitor tolerant RT-qPCR mix that we could find off the shelf. So I basically went out there, I bought whatever I could lay my hands on, and I ranked them in order of inhibitor tolerance. Now, that competitor mix became our benchmark. It's important to know that most of the inhibitor tolerant mixes out there are not lyocompatible at all. So after a few leaps in the generations of our reagent formulations, we did a comparison to the best rank of the shelf mix. Now, this is shown in green. The undotted full lines are LEX in blue and competitor in green. Now, this is in clean, without any inhibition. We can clearly see improved performance with the LEX lyophilized reagent mix in comparison to the lyo-uncompatible competitor mix. Also, in the dotted lines, the LEX in blue, the contrived, is LEX amplifying in our contrived inhibitory system. And the green dotted lines right at the bottom there is the competitor barely amplifying in our contrived system. It's fair to note that our contrived matrix is very harsh. So the ability to amplify in this background and then tying it up with development on actual human samples mean we can deal with an incredible amount of nasal sample inhibition in comparison to what is out there at the moment. Before any LEX reagent mix gets the approval for speed trials on our development platform, they have to go through an extensive inhibitor tolerant assessment using a wide range of human samples. Now, this involves spiking known viral copy numbers using a dilution series down to the limit of PCR sensitivity in the presence of a nasal eluate. Any reagent formulation basically has to display exceptional inhibitor tolerance on the benchtop platform at our LOD for molecular PCR sensitivity. So the data on this slide represents pooled human samples from 100 times to 0.1 times our LOD with amplification evidence in all series. It's important to note that we do optimize our assays to display a very high endpoint fluorescence. Now, this is very, very critical when you have nasal samples that tend to highly impact endpoint fluorescence. The development that comes with taking a LEX reagent mix from amplification in a 25-minute benchtop PCR to ultra-fast LEX speeds, it involves a huge understanding of the assay, plus the additional requirements of skilled scripting, thermal cycling development, and our assay development team, they do apply very clever strategies to demonstrate inhibitor tolerance at ultra-fast speeds. So the data on here shows one of such scripts is a 7.5-minute script, and we can clearly see that viral counts as early as 5 minutes is called with our 100 times LOD, with our 1 times LOD, our actual LOD, getting called round about the 6-minute mark. More recently, thermal cycling development continues to improve leaps and bounds. With this amplification plot shows detection of 10 times our LOD in just 3 minutes, 25 seconds, using a 4-minute, 17-second script. Important to know that this is ongoing development, and this means we continually learn of what particular reagent components that support faster cycling, inhibitor tolerance, and we tweak this to continually push PCR performance at ultra-fast speeds. Beyond our Gen 1 respiratory product, the fact that the heart of LEX is fast PCR, now this means the product has a very wide application. Some examples of other tests that are within our scope include those of strep A, chlamydia, and gonorrhea, to mention a few. Additionally, the LEX technology could be used as a central lab tool to enhance throughput. In conclusion, we at LEX are aiming for speed, molecular PCR sensitivity, and reliability by the fact that we've reduced the handling and we're increasing ease of use in a low-cost product. I would like to say thank you for taking the time to join us on this webinar, and happy to take questions.
Thank you, Andrew, for that interesting presentation. We hope that everyone listening enjoyed the webinar and found it informative. We will now be moving on to the last part of today's webinar, the question and answer session. The poll questions mentioned earlier should also now be appearing on your screen. Alongside Andrew, we also have Chris Hull from LEX Diagnostics joining us for this session. As a reminder, please feel free to continue asking those questions using the tab at the left of your screen. Simply hover over the speech bubble icon and click to ask your question. So, Andrew, the first question I have for you today asks, what advice would you give to those starting the journey on assay development?
That's a loaded question. Advice, I would say proof of principle is key. So you need to have a proper feasibility study. You need to know your plans for commercial success. You've got to prove that the assay actually works. Also, most people also miss this point. There has to be market demand. There has to be market demand before you embark on developing any assay. Then you have to plan for commercial success. Then comes in the scientific bit, which is optimization, development, reagent, assay, shelf life, and VMV, etc.
Brilliant, thank you. Our next question for you. Does your sample conditioning strategy work beyond enveloped viruses? For example, other virus types or gram-positive or gram-negative bacteria?
That's a good question. So it's key to state here that in addition to exploiting the fragility of the envelope viruses in our blister solution that I mentioned in the slide, it's important to note that the design and the constituents of our chemical conditioning strategy, this was coined from years of extraction expertise. However, I have to state that we haven't evaluated non-envelope viruses in detail. That is not the current focus of our Gen 1 product.
No, brilliant, thank you. A sort of follow-up question. Phrased as, one hears the phrase low cost used quite often in point of care testing. Audience member asks, what is low cost in terms of your perspective?
Okay, so the premise of LEX, we're looking to displace the antigen market. So for a sale price, we are aiming for the mid to low teens in terms of a sale price at scale. And that is a ballpark of where we want to be. in terms of the ability to displace antigen tests currently in the market.
Brilliant. No, thank you. The next question, what have you found is the sort of key when you look into supplier selection?
Supplier selection. Okay. So in terms of LEX and what we're trying to achieve, what I was looking for in the first instance was price. So a supplier that would give us our COGS at scale. Quality, it also has to be a supplier that is really focused on quality of the products. They have to be very reliable. Now, when you're doing a fast development like what we've been able to achieve in 2 years in LEX, you need the technical people in that company to be very flexible. So in the case of LGC, we've had quick turnarounds. They're able to supply us custom solutions to products that they might not necessarily have on the shelf, and this has been very critical to what we've been able to achieve. Also, technical advice from the supplier, because the supplier knows their reagents in this instance better than you. So it's always key to have that technical support. It's very important.
No, brilliant. Yeah, someone who can sort of work on the ground with those tight windows, I can imagine. Lots of questions coming through. The next one, could your reagent technology be used for any application of PCR that exists today?
In technology terms, yeah, we can. There's scope. We can do agriculture, we can do veterinary, we can do environmental monitoring. But however, we are focused on humans because there's more demand for speed in terms of human point of care testing, as most of these other areas are happy to send their stuff to central labs. So yeah, it is applicable to other areas, but yeah, our focus is currently human point of care testing.
Right, okay. We have sort of follow-up question to the previous one, talking about suppliers, and they ask, what advice do you have for someone looking for lyophilization compatible reagents?
Oh, okay. In terms of lyophilization compatible, so glycerol free is always an advantage. You would want a supplier that, like I said in the other question, which is a follow-up as well, someone is willing to develop custom solutions. You need stable reagents, at least in the wet mix format, because even before you start your lyophilization, you're going to prepare things on the bench. You would want reagents that are tolerant to at least room temperature stability. Yeah, and it's very, very important as well to have that technical support, which is very, very key to what we've been able to achieve.
Great. Next question that's come through. Will you have this technology available with custom primers?
Custom primers. So we are designing our own primer. So we do everything we've done on our cartridge and our consumable is from what we've done in-house. So we've designed our primers, we've designed our probes, just to be able to, because what we've learned in this journey is there are certain demands when you're designing primers and probes for fast PCR cycling, I mean ultra fast PCR cycling. So we went blind, but we're learning as we go along what kind of criteria that is needed. So our primers and probes are novel and they're suited to fast PCR cycling. And it's from trial and error, it's from learning, and it's from having different criteria that we think support fast PCR cycling.
No, thank you. The next question, actually sort of expanding more holistically on those criteria, asks, what are your criteria when you're designing the ideal PCR diagnostics product?
Okay. So the ideal PCR diagnostic product, so I'll talk about this in terms of point of care, right? So in terms of LEX, we're looking for actual proper speed, right? True point of care. We want to eliminate manual handling as much as possible. You want your instrument or your equipment to be very small, lightweight, take up less space on the bench. And then in terms of the assay, in terms of the reagents, you want it to be affordable, sensitive, and specific in a system that is easy to use as well.
Great, thank you. Yeah, sort of bench real estate being crucial in these days, I know. The next question sort of interested whether your test has a multiplex or capability, or indeed, an internal control.
Yeah, I'd say our SARS assay is a duplex assay. So we're detecting two different regions of the SARS genome in our assay. We also have an internal control, so like a sample processing control, so we know when a swab has actually been taken properly. So we have a sample adequacy control in there.
Brilliant, thank you for answering that one. The next question that's come in, asking whether your mix is adaptable to urine for things such as STI testing.
Our focus is not currently on STIs, but we know it's common knowledge that urea is the major PCR inhibitor in urine, and preliminary testing was promising, but we've not really focused in depth on STI and urine testing at the moment.
Yeah, thank you. Lots of questions coming through, so thank you, Andrew, for your time. The next one I have asks, when do you plan on having sensitivity and specificity data with clinical samples available?
Now, this is a question for Chris. Yeah, I'll let Chris take this one. Chris is our VP commercial.
Yeah. So we're going to obviously clinical trials as soon as we can. We've had some early clinical trials done. Results look good, and we got to go out for larger trials, this year and next year.
Great, thank you, Chris, for chiming in there. Appreciate it. The sort of next question, separate question, asking about the shelf life. What shelf life can you achieve for the reagents in the cartridge, and why does it have to be that long?
Yeah, so when we're designing our reagents and also the consumable, we are aiming. We know that flu seasons start November, December, and maybe just before the winter runs out. So we're designing our reagents to last longer than the normal 12 months that competitors do. So we're aiming for 18 to 24 months, preferably. That's what we're aiming for, so that a cartridge or a consumable can be used between 2 flu seasons, for instance. That's the idea.
Yeah, and understandable. Sort of a focus question on your cartridge design, asking, how robust is the blister pack on the cartridge? Obviously, having some personal experience here, they say that obviously some point-of-care manufacturers use blister packs for calibration materials, and a big customer frustration is sort of pre-burst errors. So yeah.
Yeah, so we use this not much I can disclose here, but however, we know, we are aware of the problems when it comes to pre-blister burst. But yeah, our cartridge is designed, the way it sits in the chamber is designed to really protect that blister until you hit start on the machine. So that's how we take care of it. So in terms of how it sits in the unit, we've thought that out, and the unit has been designed to protect that in the first instance.
Yeah. Great, thank you. I know you sort of spoke about your sort of clinical focus there, but a question does ask, have you explored non-primate sort of preclinical applications?
No, we've not. No. So our Gen 1 product is going to be, like I said, the influenza and SARS and the sample control. So we're just trying to be focused on that for now, and then all these will come later. Yeah,
no, great. I say time-wise, we probably got sort of a few more, 2-3 more questions, I'd say. The next one, Andrew asks, does the kit only use a swab type of sample?
Yeah, we're going to be, it has to be, because our swab port is going to be designed to use a particular swab. Yeah, so it will be a swab designed to fit in. It will be something off the shelf. It won't be anything weird or anything new. Something off the shelf. Yeah.
Okay. And what is the limit of detection in terms of your copy number?
Yeah, so we're aiming for, there's not much I can say in terms of limit of detection. What I can say is we're aiming for molecular PCR sensitivity. So we're not looking to reinvent the wheel here. We're just looking to make our assay fast enough and sensitive enough in the presence of actual nasal inhibitors at speed. So we're not looking to invent the wheel. So it will be compatible. It will be comparable to what is out there at the moment. So we're not looking to invent the wheel. But yeah, at the moment, it's really, really very good.
Great, thank you. Final couple of questions then. Do you have any issues with GC-rich pathogens?
No, we've not. Like I said earlier, we've not really been investigating. So all the areas we've been designing, there's not much I can say in terms of IP. Yeah, but we know. Yeah. So in terms of GC rate, we are fine. No, there's not much I can say about it. I'll just not say much about that. No, yeah.
That's all right. Thank you. Final question then. Can your PCR mix be used for PCR and other PCR machines like the Quant Studio?
Yeah, we actually start development on the Quant Studio. So our development starts on the Quant Studio. We set it to the fastest speed it can cycle, and then we transfer it to the LEX fast PCR system. So we've used light cyclers, we've used a lot of standard benchtop machines. So yeah, it is compatible.
Great. I'm afraid that is all we have time for today. Thank you again, Andrew, for all of your time, as well as the discussion, of course, your presentation and this Q&A session. And a big thank you for everyone joining us online for submitting your questions. We hope that you found this a worthwhile session. If you have any other questions, please feel free to email myself at editor@selectscience.net, and we'll be able to follow up with these after the webinar. If you'd like to listen again to today's webinar, or perhaps invite a friend to listen, it will be available to watch on demand in just a few days' time. Goodbye, and thank you once again for joining us.
Thank you very much.